So what we've got here is a plasma membrane sitting right here. Plasma membrane is about 5 nanometers thick. Tiny, tiny thing. And then here we've got the cytosol. And here we have extracellular fluid. That's good. And you'll notice that there's a set of channels in the membrane. These are potassium channels, and they're going to allow potassium to flow from one side to the other. So let's look at the distribution of potassium on the two sides. You'll notice the concentration of potassium is much higher on the inside than on the out. It's about a 40-fold difference in potassium, because the inside sits at something like 140 millimolar, and the outside sits between 3 and 4 millimolar. So a 40-fold difference in potassium concentration. And that's balanced by a set of organic anions. So these are proteins that are negatively charged a lot of the time because they have attached to them phosphate groups, which have a net negative charge of -3. So all these organic anions in here are heavily charged negatively. So there's some chloride, but the chloride concentration is actually about more than 10x greater on the outside than the inside. And then the sodium concentration is also about 10x greater on the outside than the inside. Calcium's missing from here. Calcium, because it doesn't have a lot to do with the resting membrane potential. Calcium has a 12,000 fold greater concentration outside than in. All right, so what happens? We've got way more potassium on the inside than the out, and these channels right there, the two of them are called leak channels. They're ungated. So unlike the channel we saw in a previous video, these channels don't have a gate to them. They're constantly open, and as a result, potassium flows down its concentration gradient. It goes from a place of higher to lower concentration. Now, as it does that, it carries a net positive charge out of the cell and moves it into extracellular fluid, and it leaves behind a net negative charge because this organic anion now doesn't have, at least one of the negative charges on it, doesn't have a potassium to buffer it. So as a result, negative charges build up. And that process whereby potassium leaks out of the cell makes the extracellular fluid a bit more positive and the cytosol a bit more negative, leads to the generation of a resting membrane potential, what we usually call resting MV. Resting VM. The resting membrane potential, so voltage membrane, the resting membrane potential, which is usually somewhere between -65 millivolts to -75 millivolts. I typically use -70 as sort of as a starting point. So we're going to have this resting membrane potential where the inside of the cell is negative relative to the outside. We're always going to use the extracellular fluid as ground. We're going to count it as being 0 for just about everything. We're going to say, okay, the outside, we're going to hold it as a constant, and we're going to now look at the difference in charge between cytosol and extracellular fluid. And when we do, we find cytosol is negative, and it's negative by a value of something like 65 to 75 millivolts. So minus 65 to minus 75 millivolts is the resting membrane potential. And that occurs because potassium is allowed to leak out of the cytosol and move into extracellular fluid.